Hello students, welcome to zoology class. Hope all of you are fine. Today, in continuation to my presentations on cell structure and function, this is my second presentation on cell structure and function. Today I will talk about the ultrastructure of eukaryotic cell. As all of you know that there are two types of cells, prokaryotic cell and eukaryotic cell. Prokaryotic cells, they are primitive cells, and eukaryotic cells, they are developed cells. And we will study in this presentation about the ultrastructure of eukaryotic cell. So the eukaryotic cell, it is present in all the animals and plants. So eukaryotic cell, it is composed of a jelly-like transparent protoplasm which is bound by the plasma membrane. And protoplasm, it has two parts, cytoplasm and nucleus. As you see this in this flow chart, that cell, it has two parts. One is the protoplasm, and protoplasm is covered by the plasma membrane. And then this protoplasm, it has two parts. One is cytoplasm and other is the nucleus. And this cytoplasm, it has compartments inside it. So some of the compartments, they are membranous, that they are surrounded by membranes, and some compartments, they are not surrounded by membranes. And these all compartments, they are known as organelles. And the membranous organelles means the organelles which are surrounded by membranes. They are endoplasmic reticulum, then chloroplast, mitochondria, and Golgi complex. And non-membranous organelles are just like as centriole and ribosomes. So this is about the basic structure of the cell. So we have to study about these cell organelles one by one. And so today we will start with the cell membrane or the plasma membrane. Plasma membrane is also known as the cell membrane. And we have to study the structure of cell membrane or the plasma membrane and its function in this presentation. So after the discovery of electron microscope, the cell membrane structure was studied in detail. And eukaryotic cell, it is compartmentalized by a number of organelles, as I have already told you, and these organelles are bound by biological membranes. And so these are the biological membranes, and the plasma membrane is that membrane which covers the whole of the cell, that is known as the plasma membrane. And the membranes which cover the organelles within the cell, they are known as the subcellular membranes. So there are two types of biological membranes. One is the plasma membrane which covers the cell, and the subcellular membranes, they cover the organelles within the cell. So now we will come to the structure of the plasma membrane. So when we talk about the structure of the plasma membrane, many models have been given describing its structure. So first structure or first model for the structure of the plasma membrane, it was given by Daniel and Debson. So it is known as the Daniel-Debson model, and it was given in 1935. And according to this model, the plasma membrane is made up of three layers. And what are those three layers? One is this is bimolecular lipid layer, or it is the bilayer of the lipid. One layer is this, from this point to this point, and the other from this point to this point. So these are two layers of phospholipids, or you can say simply lipids. And these two lipid layers, they are sandwiched between the two layers of proteins. So this is the proteins coat on the external side of the cell membrane, and this is the protein coat on the internal side of the cell membrane. So we can say that according to Daniel-Debson model, the plasma membrane has three layers. One is the bimolecular bilayer, which is a lipid layer, this one, and it is sandwiched between the two layers of the proteins. One protein coat is this and the other protein layer is this. So lipid molecules are set at right angle to the surface. So this is the surface and lipid molecules are arranged at right angle to the surface. This is surface and this is lipid molecule. So they are at the right angle. And the total thickness of the cell membrane, it is 8 nanometer according to Daniel-Debson model. And this lipid layer from here to here, it is 6 nanometer, and this protein coat is 1 nanometer, and this protein coat is also 1 nanometer. So it comes to 8 nanometer. So this is about the Daniel-Debson model of cell membrane. So next model, it is the Robertson model. This model was given by Robertson in 1965, and this model only confirms the Daniel-Debson model that the cell membrane, it is made up of three layers, that is the lipid bilayer and the protein coats on either side. So according to this Robertson model, all biological membranes, they have a trilamellar structure, means they are made up of three layers, and three layers, they are proposed in the same way as they were proposed in Daniel-Debson model. But according to Robertson model, the thickness of the cell membrane is 7.5 nanometer. So we can say that this concept of Robertson model, it is an oversimplification and it does not give account for many proteins. So this is the Robertson model, and this is just like Daniel-Debson model. And the next model, it is known as the fluid mosaic model of cell membrane, and it was given by Singer and Nicholson, and this model was given in 1972, and it is known as the fluid mosaic model. So this is the most favored model, and this model is described as protein icebergs in a sea of lipids. So these are the proteins and these are the lipids. So we can say, according to fluid mosaic model, protein icebergs in a sea of lipids. So this model is described as protein icebergs in a sea of lipids. So these are the lipid layers, again double layer of lipids. This is protein layer and again this is protein layer, and these are also the iceberg like proteins. So exactly it looks like the protein icebergs in a sea of lipids. And this, according to this model, the membrane associated lipids, they are amphipathic, means they have polar and non-polar ends, and so they contain both hydrophilic and hydrophobic ends. Polar ends interact with water, so they are known as hydrophilic. Suppose this is the outer side of the cell membrane, so these will be the hydrophilic ends because they interact with water, and the non-polar ends which do not interact with water, they are the hydrophobic ends. And so outer surface of the membrane is hydrophilic, and the inner or the anterior surface of the membrane, it is hydrophobic. So this is about the fluid mosaic model. So there are three models given by different scientists about the structure of the cell membrane. One is the Daniel-Debson model, second is the Robertson model, and third one, which is more explained, that is the fluid mosaic model. So this is all about the three models of the cell membrane. Then comes the chemical composition of the plasma membrane. So of which constituents the plasma membrane is made up of. I already talked about you that the plasma membrane is made up of lipids and proteins mainly. So lipids are 20 to 79%. Proteins are also present in the same proportion. There are also present oligosaccharides. They are about 1.5% of the total component, and water is about 20%. So this is the chemical composition of the plasma membrane. Then comes the types of proteins which are present in the plasma membrane. So these are the three types of proteins which are present in the cell membrane or the plasma membrane. Number one is the peripheral or the extrinsic proteins. These are present on the surface of the membrane and they are loosely connected, and so they can be easily removed in aqueous solution, means in solution of water, and they have chains of sugar attached to it. So these are the peripheral or the extrinsic proteins which can be removed easily in the aqueous solution. The second type of proteins, they are known as the integral or the intrinsic proteins. These proteins, they are embedded in the bilipid layer. As you know the structure of the cell membrane, there is the bilipid layer, and these integral or the intrinsic proteins, these are embedded in that bilipid layer and they cannot be removed easily, and they give shape to the membrane. And these proteins, that is integral or intrinsic proteins, they can act as carriers or channels or receptors. When they act as carriers, they transport molecules to and fro for the membrane, and then they act as channels. Then they passively transport molecules across the membrane, and when they act as receptors, they act as single molecules and transport information into the cell. So these are the intrinsic proteins. And another third type of protein which is known as transmembrane protein, and these proteins, they form the major component of proteins in the cell membrane which float on or in the bilipid layer, and these are the glycoproteins. So these are the three types of proteins which are present in the cell membrane, which form the structure of the cell membrane. Next comes the transport across the cell membrane, that how molecules they transport across the cell membrane from inside the cell membrane to outside the cell membrane. So there are two types of transports across the cell membrane. One is active transport and the other one is passive transport. So when we talk about the active transport, it is that type of transport when molecules move across the membrane with the use of energy. That is, when they use energy to cross the membrane, that type of transport, it is known as active transport. And few molecules are transported across the membrane against the concentration gradient, means from lower to higher concentration. So when the molecules move from lower to higher concentration, that is against the concentration gradient, then it is the active transport, and then energy is used, and ATP is used to get the energy. ATP is a source of energy, and the example of active transport is the sodium potassium pump. So this is about the active transport. Then when energy is used to go across the cell membrane for some molecules, then it is active transport, and energy is used because it goes against the concentration gradient, that is from lower to higher concentration. And then comes the passive transport. And what is passive transport? When the molecules move across the membrane without any use of energy. So then it is known as the passive transport. And passive transport it can occur by means of three ways. I can say that passive transport is by three means or three ways. What are those ways? They are diffusion, then osmosis, and the third one is facilitated diffusion. So passive transport across the cell membrane occurs by these three means, that is diffusion, osmosis, and facilitated diffusion. First of all, what is diffusion? It is a process in which the molecules of the substance from an area of high concentration move across the membrane to the area of the low concentration. So this is the cell membrane. This is the area of high concentration, that as I have shown many solute particles here, and this is the area of low concentration. And when the molecules move from this place to this place across the cell membrane, that is known as the diffusion. That is from higher to lower concentration. These molecules are moving from higher to lower concentration. So this is known as diffusion. Second way of passive transport across the cell membrane, that is known as osmosis. And what is osmosis? The movement of water molecules through the selectively permeable cell membrane or the plasma membrane is known as osmosis. See here, so this is a semi-permeable membrane. Semi-permeable membrane means that some things can go across these membranes, and some things are not, certain things are not allowed to cross this membrane. So this is semi-permeable membrane. And when the molecules of water, they move from this place to this place, that is known as the osmosis. So the osmosis is the movement of water molecules through the selectively permeable membrane. So this is osmosis. Then the third way of passive transport is the facilitated diffusion. So when the polar molecules, they cannot pass through the non-polar lipid bilayer, they need some carrier proteins of membranes to facilitate the transport across the membrane. And this type of diffusion with the help of the carrier, it is known as facilitated diffusion. See here, so this is the area of high concentration, this is the area of low concentration. In case of diffusion, there was no need of any carrier molecule, but in facilitated diffusion, these are the carrier molecules. They carry the molecules and then they cross this cell membrane. So this type of transport or the passive transport is known as the facilitated diffusion. So these are the three types of passive transport. So this is the passive transport, that is through diffusion, through osmosis, and through facilitated diffusion. So this is about the structure and the transport of cell membrane. Then comes the functions of plasma membrane. What are the functions of plasma membrane or the cell membrane? So the plasma membrane, it keeps the cell intact. So it is a protective barrier. Then it regulates transport of the molecules in and out of the cell, because I told you that the cell membrane or the plasma membrane, it is selectively permeable. Then small lipid soluble molecules, for example oxygen and carbon dioxide, they can pass easily through the cell membrane or the plasma membrane. Water can freely cross the membrane. So water can cross easily through this cell membrane or the plasma membrane. So we can say that it keeps the cell intact and it is the protective barrier. Plasma membrane also allows cell recognition. So it separates one cell from the other, and ions and large molecules cannot cross without assistance. So they can cross with the facilitated diffusion type of transport. And then cell membrane provides anchoring sites for filaments of cytoskeleton, and cell membrane provides a binding site for enzymes, and interlocking surfaces bind cells together. So cell membrane, it is the junction between the two cells. It forms a junction between the two cells. And then inside the cell membrane or the plasma membrane, there is present the cytoplasm, which is the fluid in the cell. So these are the functions of the cell membrane or the plasma membrane. So today we have discussed about the structure and function of the cell membrane or the plasma membrane. Now you know that there are different models given by different scientists about the structure of the cell membrane or the plasma membrane. They are: number one, model of Daniel and Edson, and second is model by Robertson, and the third is the fluid mosaic model by Singer and Nicholson. So these are the three models. Then we studied about the types of proteins which are present in the cell membrane, and you know basically cell membrane is made up of proteins, lipids and oligosaccharides, and moreover water is also present in the plasma membrane. And transport across the cell membrane, it can be active or passive, and active transport uses energy, that is against the concentration gradient movement takes place, and in passive transport no energy is used. And in passive transport there are three ways of transport: one is diffusion, second is osmosis, and third is the facilitated diffusion. Then we studied about the functions of the cell membrane, that it acts as a barrier, it acts as the junction between the two cells, and it helps in transport of material from inside to outside or from one cell to other cells. And inside the plasma membrane or the cell membrane, there is present cytoplasm, in which many organelles are also present. Some organelles are membranous, that is they are bound by membranes, and some organelles are without membranes. So this is all about for today. Now you have to go through this presentation. I have already provided you with the study material. You have to prepare notes by consulting both the notes as well as this presentation, and then in case you find any difficulty or you have any doubts, you can note them down and then you can discuss in our online class. Thank you.